Hello everyone, I am Katherine Seton from the Quadram Institute. I am a second year PhD student and Fiona Newberry is also a second year PhD student. So, at the Quadram Institute we have a theory, and this theory is that there may be an intestinal viral infection that alters the gut microbes, and this could lead to an increase in bad gut microbes that can lead to gut inflammation, and if there's inflammation this could lead to a leaky gut, and then if there's a leaky gut there could be the chance for gut microbes to escape from the gut and cause an abnormal immune response to gut microbes, ultimately leading to autoimmunity. So this is just a diagram to show a healthy gut versus an unhealthy gut based upon our theory. So as you can see, there's an intact gut barrier. There is a diverse population of microbes within the gut, and there's no immune response. Whereas, as you can see in the unhealthy gut, there is reduced variety of microbes in the gut, and the intestinal barrier is leaky, and there's an immune response within the blood. So, my actual PhD aim is to look at the leaky gut and see whether this leads to the gut microbes escaping into the blood and causing an abnormal immune response to the gut microbes, ultimately leading to autoimmunity. So to test this, we have begun a human study at the Quadram Institute where we're recruiting severe ME patients who are either housebound or bedbound, and we have 2 identification centres. So the first is through occupational therapists in Norfolk, and from these we are recruiting Norfolk and Suffolk patients, and we also are recruiting through Dr. Amalak Banzel in London, and as you can see there's quite a wide area that we have to travel to actually get the samples. So we are looking to recruit 10 pairs of severe patients and household controls. And a household control is someone who is either related to or not related to the patient, living with or caring for the patient. And it's really important to have household controls because the environment has a massive impact on the gut microbes in our intestine. So by using household controls, we're hoping to be able to identify disease-specific alterations. So the experimental approach that I'm taking is to collect blood samples and stool samples. From the blood samples, we will be isolating both immune cells and antibodies, and then from the stool, we will obviously be getting the gut microbes. And I will be using a number of techniques to see whether or not the immune cells and antibodies can react with and bind to the gut microbes. So far, I have developed 2 techniques, and using the patients that we've recruited to date, I have found that the severe ME/CFS patients actually have a significantly higher level of gut microbes that are recognised by antibodies. So if you're interested in following my research updates, you can follow me on Twitter. I'm quite new to Twitter, but I'm trying to interact with the public. So thank you. It is now over to Fiona, who's going to explain Ernie's work first, before her own.
Yes, thank you.
Good morning, I'm Fiona. I will do my best to try and explain Ernie's work, but any difficult questions, I will give you his email address, and I'm sure he'd be delighted to answer them. So, as Katherine talked about the microbiome, how exactly do you get the viruses out of a fecal sample? Well, obviously it's quite messy. You start off with the fecal sample, and we go through various steps of virus isolation. So it's basically a coffee filter, is what I'm saying. And out of the end, you get viral DNA, which is then put on a sequencer, and it tells us what viruses are in there. So this is the main part of Ernie's project, is developing this protocol so it's standardized for all of our patient samples. The next part of his project is to determine what exactly these viruses are doing. Are they altering the microbiome. The way he's doing this is by isolating specific viruses from the feces and then determining if they kill the bacteria or not. The ones that do have an effect on the bacteria, he will then be putting into animal mice and determining if these mice exhibit some MECFS symptoms, which he'll be doing this through exercise trials with the mice, which doesn't really sound very pleasant for the mice. So this is my PhD project. I'm a second year PhD student, and the main part of my project is to identify microbiome and virome alterations in severe MECFS patients. So me, Katherine and Ernie, we're all using the same patients. So the main question is, what happens after you've isolated the viruses from the feces? They go on a machine that tells you what is in there, but you need to interpret the results using computer programs. So I've had to learn computer code for my PhD project, and I never thought I'd be looking at a command line on a computer. But there are several problems associated with interpreting the results that you get from the viral microbiome. Viruses are highly variable. They're one of the most abundant microbes on the planet. They're constantly changing. As you know, when you get the flu kind of travels around everywhere, and it mutates as it changes through the population. Only a small fraction of these have been identified. So in a lot of the fecal samples, we have no idea what the viruses actually are. And the tools that are used for analysis are massively underdeveloped. So this side of the workflow is Ernie's PhD project, so he gets to deal with the messy stuff, and then I get the nice computer stuff at the end. My part of the project is developing the programs that are needed to interpret this, and obviously it had to be a Mac, I wouldn't use a Windows. And then kind of determining, is there a difference between MECFS patients in terms of their microbiome and their virome? If there is a difference, what does this mean in terms of the functionality of the microbiome? So, is that the last slide? I think it is, yeah. Thank you for listening. Are there any questions?
Okay, are there any questions? It's exciting to see novel work being carried out, and who knows what's going to turn out. But, you see the charities invested in studentships, and interesting, Give them some questions, please.
I'm always curious about how one tests mice. The Chinese do this extensive exercising where they have them swimming massive laps. And my quibble is that doesn't replicate ME. ME is exhaustion from the inside out.
Yes. Yeah.
And I want to address that to your absent colleague.
Yeah, well, when I get back to Norwich, I will definitely be saying to him, why did you abandon me with this question? To be honest, that is something that is Ernie's side of the project.
Yeah.
I know that the various tests are going to be involved, and as you mentioned, that isn't MECFS. I think he's going to be combining it with other kind of tests. He's not going to say that the mice have MECFS. It's going to be more they're exhibiting fatigue symptoms. Sorry, I can't answer that very well.
My question, Ian, is this.
Where are you? Oh, there you are, Malcolm. Yeah.
Yeah. The problem is, I think, first of all, you're very brave. I think, secondly, you'll get new data, so your PhD is assured. You'll get new data, whatever you touch, you'll get new stuff. So that's good news for you. But the problem I have, and that you're going to have to face, really, is there are now factors influencing the microbiome in everybody's diet. And this, I'm thinking particularly of glyphosate, which is in everything we eat. And you're getting a persistent low dose of glyphosate. Everybody is. We're all getting it. And we don't know what that will mean in terms of cumulative effect on people's health. But it's there, And glyphosate chelates metals, it kills bacteria, it kills the good ones in the gut better, quicker than it does the bad ones, and it's got some suggestion that it might be virucidal.
Yes.
So just keep an eye on it. Just be aware of it, really. Norfolk is well known for its agriculture, so there might very well be quite a bit of it around. Even in population, you might think it should be healthy. Yeah.
Yes. There are other people in the audience too who are doing similar work to this, and they're very welcome to say something. Fiona, do you want to answer that?
Well, that was a very long-winded question. I'm going to just take part of
the way to tell them.
With the diet, we are actually taking food diaries so that we can record and see if there's any differences between the patients and the household controls, because obviously they might not be eating the same thing. And we are potentially going to be using an algorithm that I am going to be asking a different PhD student for, which can kind of determine whether the diet is inflammatory or not, and then that might explain potential outliers in the results.
Keep an eye on it, that's all.
Yeah, good. Anyone else who's working on this? Yes, sir. Yes.
Yeah, hi, Ken Holdcroft from Newcastle. In your studies, have you thought about, have you had the opportunity to look at identical twins, one of which may have CFS-ME and one that doesn't, so that you have sort of a control, shall we say, of identical samples, which may show up the differences, i.e. a leaky gut, non-leaky gut? Is that a possibility that you've looked at or been able to study? Or
We'd love to.
Do you think it's a future study?
Yeah, so basically when we have household controls, we can't be like, how many family members do you have? Do you have any identical twins? So purely we have to go on and use who actually wants to take part in the study. We can't influence that whatsoever. But if we do get a twin, that would be great. Obviously, we have questionnaires for the household controls, so we say, what is your relation to the patient? So I can't actually determine and ask specifically to study them, but if we do get them, that would be absolutely amazing, yeah.
Okay. All right. Right, one last one, yes, at the back, sorry. The woman at the back, yes?
I was involved and interested in studying PhDs for MECFs.
Did you hear that?
No.
How do we both get interested in it?
In studying.
Yeah. So for me, I was looking for a PhD studentship around human disease, and then this one came up, and I Googled MECFS, and I'd never actually heard of it before. And then I was kind of shocked at what I read and how it has been taken by the medical community as being a psychological disease. And I thought, I want to study this.
For me,
That's telling them. Yeah. Good for you. Yeah.
I was actually diagnosed with ME when I was 13. So after being at university, I actually realised that maybe I could go on to study ME and try and find out what the hell is actually happening in ME. So I actually went on findAPhD.com once I finished my undergrad, and there was only 2 PhDs in ME research that were being advertised, and that was mine and Fiona's. And obviously, I would not be able to work with computers. I am a computer...
I hate immunology, so...
so it worked out perfectly because I absolutely love immunology, so yeah.
We were discussing this morning immunology. Yeah. What was it you were saying, Beth? Nobody really understands all the details and would hate to answer an exam question in immunology. We never could understand the lectures on it, so it's up there somewhere. But it's so complex, and it's very brave to do research anywhere in this field at all. I take my hat off, well done. Yeah. Cancer's too easy, that's what I did. Yeah. It's too easy, yeah. One more question,
well,
yes.
It just really was to offer you encouragement, because it's very, brilliant to see 2 young women getting involved in this, in research in this area, and to say that I think it is an area that needs exploring, and my daughter's got ME/CFS that she's had since she was 13 after the HPV vaccine, which is very interesting. And we, in desperation, we're with the private environmental doctor as well, trying to improve symptoms. And one of the areas we've been looking at is her gut, and her gut is very closely linked to the severity of her symptoms when she has them. And the finding we had was that her gut, the diversity in her gut was very low, and we're trying to increase that with probiotics, which do seem to are helping. And I just wondered if you're going to look at that sort of area, like probiotics and things like that.
Probiotic research is kind of a black hole. It's very interesting, but I don't think probiotic research by itself is there for us to study it in ME/CFS. The way to study it in ME/CFS would probably be to identify the key microbes that are depleted and which ones would be beneficial, and then maybe develop an ME/CFS specific probiotic. But yeah, it is very interesting.
Right, well, thank you very, very much. Oh, one last one, yes, please.
Hello, I'm Eva Martin. I'm the doctor from Spain. And would it be interesting to know if your patient have any intolerance to say, please?
Any intolerance before to say the alteration is for ME?
Intolerance, lactose, fructose. Yes. Do you understand me?
You get that?
Food allergies. That isn't. Do we collect it in the food?
We're collecting information on what patients eat in the hope that if they can't have gluten, they will tell us that they have gluten-free bread. If they can't have lactose, they'll tell us that they have lactose-free milk and stuff like that. But we're not specifically asking about their allergies and food allergies, or whether or not they have to exclude things from the diet.
Okay.
Can I say one final
go
thing?
on,
I forgot to say this on my slide, but thank you to the charity for funding us, and also Solve ME/CFS initiative. I was awarded the Ramsey grant, so they're helping fund this research as well.